Hi everybody, it's Mrs. Peachy from your WCA biology class, and today we're going to be looking at Unit 4, Lesson 4 of your biology portfolio's Modeling Meiosis. And so hopefully the purpose of this lab is that by the end you should understand how meiosis takes place and kind of really have a physical idea of what these cells actually have inside of them. Okay? So some of the things that are required, it does say pop beads, magnetic centromeres, a large sheet of paper, colored pencils, and scissors. You don't have to use all of those materials per se. As far as the pop beads go, they look like this, okay? And they're a little easier to use because the beads literally kind of pop together, okay? So you can kind of hear that. Making sure my volume is on. I just did a whole recording and I was muted, so that kind of stinks. You could also use Legos in place of pop beads, which works well, and a lot of people have used that. They're not flexible, so they don't model the best. And there are these things which are math blocks that I'm going to use today. Okay? How many you need and how many colors? You need 4 different colors. You need 20 of one color, 20 of a second color, so 2 groups of 10, 10 of a third color, and 10 of a fourth color. And you can use a bunch of different things. I've had people take different colors of clay and make little balls and kind of put them all together. I've had people take regular beads and string them on wire or string, and that works too. So there's lots of options for you guys. You just have to be a bit creative. But to answer your question, yes, you do need to make a 3D model of these cells because it's really the most effective way to learn about them. There are some questions to start, and they call them pre-lab questions. One of the questions is, why must you use the same number of beads when you construct the second chromosome? We'll talk about that one. Question 2 is, infer, why is the longer chromosome used to model crossing over? Talk about that one too. And the calculations we'll kind of do at the end, because those tend to be the thing that tricks people the most, becomes the most tricky. All right, so now we're going to go ahead and do the rest of this modeling on my desktop. So hopefully you'll be able to see that, and it'll be kind of easy here. So I'm going to kind of swing this down and flip it upside down. And I've tried this a number of different ways, but I'm going to try using a roll of paper toweling because I think that might work really well. Okay, so this is my large piece of paper. All right, so first of all, to think about the idea behind making or what these cells are. These are your reproductive cells, egg and sperm, and they contain half the number of chromosomes as regular body cells, regular somatic cells. So you have what are called gametic cells. Okay. And they're haploid. Ooh, look at these fun vocabulary words. Haploid, remember, means it contains half the number of chromosomes. And your regular body cells, like the cells in my skin here, would be somatic cells. S-O-M-A-T-I-C, somatic. And they would be diploid because they would contain 2 copies of each chromosome. Okay? So in our fictitious organism, they only have 2 chromosomes total, 2 copies of 2. In our bodies, we have 23 pairs of chromosomes, so we have a lot more, but we're going to simplify it so that it's easier to kind of follow, I guess. All right, so say this is our first chromosome. Notice that I have 2 different colors here, and that's just so that we can easily kind of keep track of the fact that this one, say, down below, came from mom. And this one came from dad. So they have the same genes in them. Each of the blocks represents a gene. So let's just call this gene O. I'll just use this pen. I'm writing all over these now, so they're getting to be a little messy, but so we'll call this gene 1, 2, 3. 1, 2, 3. Kind of helps us just kind of keep track of things a little bit, just so you kind of get an idea here, right? And then according to the lab, you have a second chromosome over here that's a little shorter. Okay. So I'm just going to get rid of that guy, because it becomes just a little bit too much going on to be able to pay attention to that. You have to understand that, yes, this one's here too, okay, but we're just not going to look at it right now. The first process of what occurs is that these pair of chromosomes is going to make a copy of itself, okay? And I guess I called this one Dad this time, so that's what we're going to do. So now we got Dad. Now before I forgot to mention is that, like, when you look at one single chromosome here, these little pieces have the same genes, booger. So they're called sister chromatids. And I know it's kind of funky that it's mom and dad and sister, but just kind of go with it, okay? So here, these are sister chromatids. Now that they've got two identical kind of copies of each other, these two are called homologous chromosomes. So they're the same, but they're copies of each other. And that's important because later on, when we go through Meiosis 1 and Meiosis 2, you'll see the difference. Okay, so this happens prior to meiosis. So the first phase of Meiosis 1 is called Prophase One. Stop it. This, by the way, works a lot better without, with the beads. And if you had the magnetic centromeres, you wouldn't have all this kind of bumbling and fumbling. So this is Prophase One. One kind of cool thing that happens during Prophase One is you get all of these sister chromatids. They get kind of all jumbled together, and you get this big structure that's made, and all four of them are all crisscrossing over each other, and this is called a tetrad. And when this happens, the chromosomes get kind of tangled, and you get an effect called crossing over that takes place. Okay, and when crossing over happens, what you might have is you might have like a chunk of this gene, or a chunk of this chromatid come off, and these genes kind of go over here, and these genes go over there, and you'll notice that you still have them in the correct order. It's still 1, 2, 3, 1, 2, 3, but now they're on the opposite. They're on the sister chromatid. Okay, so that happens during Prophase One. That's an event called crossing over, and it increases genetic diversity. So then, that's Prophase One. The next stage is Metaphase One. And during Metaphase One, these sister chromatids still stay attached by their centromeres, and they line up at the center of the cell, like that. Okay, but they're still attached in the middle. So if I had little magnets, they'd be magnetically attached, and in real life, that's not magnets, but they're still attached. Then, that's Metaphase One, because they're in the middle, remember? Meta means middle. And then during Anaphase One, one of these sets migrates north, one migrates south, okay? And if I had my other shorter ones over here, you would get one of these also going north, one of these also going south, okay? In the human, you'd have 23 going north, 23 going south. Just too much stuff going on there, though. And during telophase, the new cell membrane forms, and you have two new cells, each with a single set of chromosomes. So each one is now diploid. But we don't want them to be diploid, remember? We want them to be haploid, so we still have to go through Meiosis 2. So Meiosis 2 starts out with this new membrane dissolving. Now you're at Prophase 1, because these all kind of come together again. And, oops, excuse me, they do not come together because these are two separate cells. My bad. But we're just going to look at one cell. So we'll just look at this guy for now. The nuclear membrane around it dissolves. These guys are in a separate cell and they're doing the same thing in the separate cell, okay? So the nuclear membrane around it dissolves, and now the centromere becomes detached. And in Metaphase 2, these guys are going to line up in the center of the cell just like that. And now one will migrate to the south and one to the north in Anaphase 2. And in Telophase 2, a new cell membrane forms. Now, if you can imagine, we don't just have two cells like this. Come here. This is so technical. I just, the special effects are kind of eerie, aren't they? You actually get 4 cells that are like this. And remember, we had those blue and white guys too. So those get part of it. So you end up with 4 cells, each with a single chromatid. Okay? And each one now has half the number of chromosomes that the original body cell contained. They are haploid, is what that's called. Okay? And notice how jumbled up they are. They might get a white one, they might get a blue one, they might get a red one, they might get a green one. They might get a mixed one because of the crossing over that took place. So the process of meiosis and this process, which is called independent assortment, means that there's a lot of different possibilities as to what the gametic cell might contain. Okay, so this is going to bring us to some of those questions on the board. And we're going to go to that here. All right, so hopefully I'll be able to go through some of the math here with you, but we just have a couple of minutes. So that question 3 says a diploid cell has 2 pairs of homologous chromosomes. How many different combinations of chromosomes could there be in the gametes? What you have to think of is that every time you have 2 pairs, right? So you're going to have whatever the number of pairs is going to be the exponent in this case. And you're always going to have pairs, and pairs is 2. So if I have 2 chromosomes, if I have 2 chromosomes, it would be 2 to the 2nd power. So 2 to the 2nd power would be 4, which I can't seem to find a place to write on the screen. I don't know how to write on here. I'm trying to figure it out. So that's how you would do the math for that one. Now, if you have, let's say, 10 chromosomes, then it would be 2 to the 10th power, okay? And if you have, like human beings, 23 pairs of chromosomes, it would be 2 to the 23rd power. And that comes in handy when you go further down. This is where you would draw your pictures. And down here, it does say what would happen if you increase the number from 2 to 3. Okay, so then you would have 2 to the 3rd power. And then from 3 to 4, 2 to the 4th power. And if you were a human being, it would be 2 to the 23rd. So do the math for that and see how many potential combinations there would be for humans. And that would be without crossing over. And it's just for 2 people. So imagine, how many potential combinations there could be for all the people in the world. It's pretty incredible. So that is the end of the meiosis portfolio. If you have any questions or concerns, you will be advised to give me a call at extension 2204, or you can send me a webmail message. Thank you.